Hey y'all, thanks for tuning in. Really excited to chat with you today about evening primroses. Before we jump in, I want to acknowledge my co-authors. This work was really a collaborative project, both within USGS and across a few other state and federal agencies.
Also want to acknowledge all the folks who made this work possible, whether that was funding or site logistic information, background. Special shout out to the Park District staff, especially Browns Island, Big Break, and the Regional Botanic Garden. They were really excellent partners on this work, and the sites themselves and the populations of primrose there were pretty instrumental in our overall findings. So thank you all. So today we'll be talking about the ecology and biodiversity of evening primroses in the genus Anothera, especially Anothera deltoides, the species complex. So we'll start off talking about this beauty, federally and state endangered Antioch Dunes evening primrose, which is subspecies Howellii, named after John Thomas Howell, botanist. It's quite charismatic. It's pretty large, short-lived perennial. It has these really big, attractive white blossoms that fade to pink. You'd almost expect it to be an ornamental, not an endangered plant when you see it out in the field. This primrose is a specialist on dunes, and it's actually endemic to our estuary. Now, often we think of dunes on the coast or in the desert, but this dune is actually riverine in origin, and it's inland in the fresh part of the estuary.
If we zoom into the area, we can get a better look at what's thought to be the historical extent of this sand sheet. Now this layer is from a great report from SFEI on eastern Contra Costa County. You can see a darker brown, which is probably more shrub community. We'll remove that. And this layer is likely the total extent, at least on this side of the sand sheet, of more open, moving sand, which is the habitat that these primroses really need. Now this sand sheet was heavily developed and mined for its sand resource over the last 150 years. In fact, it was so degraded that in 1969, Jim Roof, who was then director of the Tilton Botanic Garden, wrote a memoriam for the ecosystem as it was being destroyed. That's pretty incredible to read right now, 50 years later, right? But thankfully, because of the work of the Botanic Garden, Jim Roof, and others in the area, the species was listed in 1978, a refuge was established, and at the same time, seed was brought to the Botanic Garden for conservation, and outplantings were established, including at Browns Island up here. So that led to a lot of hedging of bets, if you will, of these populations. You have the refuge here, we have a couple outplantings, and seed brought again to the Botanic Garden, which of course is far off this screen. Now the eastern side of the sand sheet is still under development today, and not from mining so much as development, especially housing. Now so far we've been talking about subspecies Howellii, the Antioch Dunes evening primrose, and that's found on the western side of the sand sheet, again at the outplanting at Browns Island and on the refuge. However, there are modern reports of subspecies Cognata, locally called the birdcage evening primrose or desert lantern further south, on the eastern side of the sand sheet, including Big Break, Regional Shoreline, Little Dune Planting, another district property. So for this project that I'll present some results on, we really wanted to better understand the distribution, the ecology, and the genetic diversity of the primroses on this sand sheet. So to do that, we undertook range-wide surveys, and we coupled that with genomic analyses. For the field side of the equation, we used systematic range-wide surveys. We set up a grid at each site. We walked transects through the grid, identified which grid we were in when we saw plants, either adults or juveniles, and we converted that to a grid system that we could combine with other data like remote sensing, NDVI, near-infrared imagery, or simple plant population data. What else is growing in the grid? We carried out these systematic surveys at all sites where subspecies Howellii has been reported or suspected over the last several decades, of course, including Browns Island. We expanded our search to find subspecies Cognata populations as well, and I have a little pink dot here for Big Break's dune planting near the parking lot there. On the genetic side, we coupled those field surveys with next-generation genomics. So at each population we visited, we collected tissue from as many individuals as we could, or up to about 20, 25 individuals. We sent those over to Dr. Liz Milano, who's working in Dr. Amy Vandergast's lab at USGS, where they developed a genomic library. If you have questions about their approach, I can attempt to answer, but I'll direct you to them for specifics. So what did we find? Well, I'll start by talking about some of the subspecies Howellii data. So how many individuals did we find at these sites? Well, at some sites we found very few individuals. They were dominated by invasive annual grasses, and there were occasional adults. At other sites, and this is a dune restoration fish and wildlife property, it was really hard to count the number of primroses we saw, especially juveniles. If we summarize those patterns, and if we combine that with some of the other ecological data we found some interesting patterns. So I'll show you two graphs here. The first one has invasion on the x-axis, and this is a good proxy in this system at least for total plant cover. So we didn't have grids that were dominated by native plants. If there was a lot of plant cover, it was due to annual grasses. On the left we have low invasion, so more of an open dune setting. On the right we have very highly invaded habitat, which is most of it, with lots of annual grasses. On the Y here, I'll show you the adult density of ADEP, which is just subspecies Howellii, the Antioch dunes evening primrose. You can see from the pattern that there's a lot more adults, exponentially more, as you decrease invasion. So in an open dune habitat, there's more than 1,000 individuals per hectare. Lots of primrose around. On a highly invaded habitat, there's less than 100 individuals per hectare. So not only did we find fewer in invaded habitat, but the demography of that population also changed. We keep the same x-axis here, but now on the y, instead of density or a number of plants, I'm showing you a ratio of juvenile to adult individuals. So here's a 1-to-1 line. So if you have a population below that line, there are fewer juveniles than adults. Not great for long-term population subsistence. Above that line, more juveniles than adults. That's kind of where you want to be for these plants. We found an even stronger relationship than with just density, where we had up to 7 times more juveniles in an open dune setting than adults, while in the invaded we actually had fewer juveniles than adults, maybe only a quarter as many. So that focused just on subspecies Howellii, but what about broader on the sand sheet? What is the distribution of primroses in this species complex? Well, as I mentioned with subspecies Howellii, we have a couple outplantings, including Browns Island to the west, the refuge. We found an additional population along the railroad tracks in Oakley. So historical distribution, likely the western side of this open sand habitat. Well, what about the other primrose I mentioned in the beginning, subspecies Cognata, or what folks locally have been calling birdcage evening primrose? Well, we found a different primrose, right, than the subspecies Howellii at Big Break Regional Shoreline, Dutch Slough Tidal Restoration owned by DWR, and a few other sites around Oakley. But we were confused with the morphology because they didn't they didn't key out cleanly as subspecies Cognata. I talked with Ryan Odell, who's an expert on San Joaquin flora and co-author on this talk, and he and I went down to Corral Hollow down here about 30-35 kilometers south of the area. This is definitely subspecies Cognata, keys out cleanly, morphology matches. We were left with a bit of a puzzle for these local Oakley specimens of Primrose found at Big Break, for example.
So that Cognata population matched the taxonomic keys and what we expected. It has flower buds that are very fuzzy, essentially, with green leaves that are not very dissected, and they are essentially annuals. They can get large, but they do not live more than a year, right? On the other end, the Howellii, they can become huge plants. They're in a more mild climate, of course. They live for several years. Their leaves are highly dissect, very hairy, so they have this gray-green foliage appearance. And their flower buds are quite a bit different. They don't have very much hair, and they have little free sepal tips at the end, which I did not think I would care so much about. But on the eastern side of the sand sheet, at Big Break and other sites near Oakley or Knightson, we found this other primrose that didn't quite cleanly fit. And one thing we were wondering is, what the heck is this thing? Is this a hybrid? Is it new? Is it old? Where does this fit in Primrose genus? So thankfully, we had genomics run on these populations from the beginning. So we had quality geneticists doing this work as we pondered this ecological distribution question. And from their work, combining all those thousands of genetic points into, in this case, a PCA plot, generally the closer things are together on this plot, the more closely related they are, or the more closely their sequences are in alignment. And the further away, the more distantly related, or the more different their genomic data are. And we saw with Cognata from Corral Hollow and from further south in the San Joaquin, they are quite distant from all of the morphologically Howellii samples. So the ones that looked like Aniactune's evening primrose, regardless of where we found them, all clustered really tightly together, and Cognata clustered tightly together from those San Joaquin sites. But all of the populations from the eastern side, including Big Break, Regional Shoreline, Dutch Slough, they all clustered together, but in their own grouping in the genetic PCA space. They were more closely related, at least on PC1, to the Aniactune's Howellii than to Cognata, which matches their geographical distribution. But they are separate, distinct. There's no overlap in those populations. There's no evidence that they're, at least recently, hybridizing or genetically connected to a separate population of evening primrose that we didn't quite know what to do with. The way to visualize these data is with this phylogeny. You can see Anothera californica, different species, as an outgroup. And apologies for the color difference, but I'll overlay the image. We have Cognata up on the top, which is a separate clade from Howellii, and Howellii is sister to this other primrose, which is pretty well supported as a distinct genetic population. So right now, we are working on writing up a subspecies description. We're calling this population that's present at Big Break and other places in Oakley as the Bay Miwok evening primrose. And that name, I'm very proud to say, was discussed and talked about with Corinna Gold and others, the Confederated Villages of Lisjan. So we've talked quite a bit about the appropriateness of that name and if it is actually an honorific. So the subspecies name, Hulpunii, is after Hulpun, or a historic village site of the Bay Miwok, which is very close to what we now call Oakley, where also what we now call Marsh Creek comes out into the San Joaquin. So hopefully in the next month or so, we'll be submitting this subspecies description for this new evening primrose, endemic to a very small area in the Bay Delta. It actually has a smaller population than the federally and state endangered primrose right next door. So once it's published, if the botanical community accepts the name, it'll be incorporated into the Jepson, etc., and all the other steps in the process of protecting this plant can start to move forward.
So to wrap up with some conclusions, for subspecies Howellii, including the Browns Island population, we supported some previous work and pushed it forward in a little more continuous framework. That open dune habitat is really necessary, especially for juvenile recruitment into these primrose populations. So on a large scale, process-based restoration, getting that open sand movement is really crucial. On a smaller scale, anything that we can do to reduce plant cover, typically invasives, right, on this sand, that will increase or potentially increase the population of these primroses. On the genetic side, we found some surprising diversity. I say surprising, but there's a lot of botanists in California, and they're good, right? So there were, from past collections in the area around Oakley, there's a lot of little comments on their plants saying, this is kind of odd, or is this integrating with subspecies Howellii? Not quite sure on the idea on this one, right? So there's this gut feeling that something strange was happening with these primroses. And we're thankful that by diving deep into the morphology and running genomic data, we're able to provide some pretty robust evidence that indeed there is something odd. This is likely an undescribed subspecies. Finally, this is a really urban and rapidly developing estuary. There's millions and millions of people. There's still diversity to be found. California is so diverse in its flora that if we look closely and we collaborate with people who do other things, ecology plus genetics plus, right, we can discover some really unique things, even still here in the San Francisco Bay Delta.
I'd be happy to start the conversation with questions. Feel free to email me. I'm now at the University of North Florida. There's my email, especially about the subspecies description I'm leading or the ecology. We had a manuscript come out. If you have questions about the genomic approach specifically, I'm happy to direct you in Amy or Liz's direction, as they led that part of this project. So I've listed Amy's email here if you'd like to reach out to her directly. Thanks so much.